Good morning, everybody. Thank you for joining us for our Precisionary Instruments webinar. And we're really honored today to host Katerina here, who is going to talk about slicing up the tumor and what she has learned from lung tumor slice cultures. So Katerina here was born and raised in Athens, Greece. So she is joining us with a large time zone. Thank you so much. She studied biochemistry and biotechnology and completed research training really globally. She was in Spain, Italy, and Scotland. Her main expertise is on basic immunology and also the aspects of the interplay between nutrition and the immune system. And she has extensive experience in explant slice cultures, which is what she's going to be sharing with us today. So right now she's working on tumor immunotherapy and using the Compresstome vibrating microtome from our company to examine the interaction between tumor tissues and autologous lymph node cells in slice cultures. So this work is importantly carried out in the frame of an EU-funded consortium, and she can talk a little bit about that soon. And the ultimate goal of the group and the consortium of mimicking the metastatic process on a chip and also to monitor metastasizing cells in real time. And with that, Katerina, I would love to hand it over to you and go ahead and start your webinar and share a screen anytime. And just a quick note to everybody, if you have questions during this webinar, please enter in the chat and I will be sure to ask your questions in a Q&A session at the end.
Okay, well, okay, so first of all, can everybody hear me properly?
Yes.
I can hear you very well, yeah.
Okay, great. So I'm not too experienced giving presentations over the Zoom platform, so I'm going to share my screen now. But just a quick question to you guys. Are you able to also see my image while I'm sharing my screen, or can you just see the presentation? So this is me sharing my PowerPoint just now, and I hope you can see this properly.
We do. We see the entire PowerPoint. I don't know if, there we go. That's perfect.
Okay, great. And from what I see, you can also see my image at the same time. And this is important to me because I even have some props here with me to show you better how I go about setting up the culture. So can everybody also see me properly?
Yes.
Okay, great. So Abby, thank you so very much for this introduction and for taking an interest in our work and inviting me to give this webinar. So yeah, like you said, I'm going to talk about attempting to slice up lung tumors with the Compresstome. And well, as you said, I'm working in the Academy of Athens. So one first disclaimer, if you guys Google this, you might come up with Plato's name, and it's not exactly the continuation of that first academy founded by Plato, because the function of that was discontinued at a certain time point. But then in the 19th century, the Academy of Athens was founded again in a newly instated Greek state. And this is the continuation of that from back then. And we are the Biomedical Research Foundation of the Academy of Athens. And as Abby already mentioned, we are working on this project in the frame of the Tumour-LNoC Consortium funded by the European Union. Tumour-LNoC stands for Tumor Lymph Node on a Chip. And so I don't know if you can see our logo here. Let me try to move this a little bit further up. So can you see our logo right now, the full logo of our project? So this is one chip. And I'm sure that when you see this over here in the logo, you can sense that we are working on something related to the lungs. So yeah, let's just start with a brief introduction on lung cancer and the metastatic process. So lung cancer is the most common cancer in men and the second most common cancer in women. This does not mean that the incidence is higher in men than women. This just means that there is one type of cancer that is more common in women, and that's breast cancer. However, it is the cancer with the highest mortality than any other cancer. And this is directly related to the fact that lung cancer is highly metastatic. These last two attributes, so the high mortality and the high metastatic potential, is particularly well, to me, it's pretty saddening if one thinks that around 85% of this diagnosis could have been spared if the beholders had decided not to smoke. So it is one type of cancer that is directly related to smoking. And yeah, around 85% of diagnosed cases are smokers or ex-smokers. So yeah, so lung cancer is highly metastatic. And there are various routes through which a tumor can metastasize to other parts of the body. So first, we have the local metastasis, the more innocent type of metastasis in which smaller tumors are growing relatively close to the original tumor. And then we have what happens when tumors are more aggressive, which is the distal metastasis. And this means that we have tumor cells growing in distal organs of the body, and usually means also a worse prognosis for the patients. Now, we have two possible routes for distal metastasis, and this is so one possibility for the cancer cells is to travel through the blood vessels, and another possibility is for them to travel through the lymphatic vessels. Now, the theory is that one tumor sheds around 1 million cells per day in the blood circulation, and when these go through the veins, all the lymphatic vessels, most of those will die, and those that pass through the blood vessels will randomly establish metastasis in distal organs if they have the potential to do so. However, the cells that are going through the lymphatics are also passing through the lymph nodes first before moving to other distal organs of the body. And the lymph nodes are a site on which it is easy for a cancer cell to establish a new niche, mainly because usually they are very, very well oxygenated. So what happens when a tumor grows is that oftentimes the tumor outgrows its own capacity of building new vasculature. When this happens, so two things might happen. The center of the tumor is largely necrotic, but then the tumor cells themselves, in an attempt to find a new niche to populate, start to travel throughout the body, like we said, either through the blood vessels or the lymphatic vessels. So tumor cells often pass through the tumor-draining lymph nodes, and they establish a new niche there before forming distal metastasis. Now let's check a little bit the anatomy of a lymph node. As you can see, it's this construction here where we have, let's say, a round formation surrounded by endothelial cells and stromal cells, which are the basis to support all the other immune cells that are to be found inside the lymph node. Here we have what we call, some cell groups called the germinal centers. And in the center of the lymph node, we have the cell zone, where the cells are to be found. In between the zones, we have the antigen-presenting cells, which are mainly dendritic cells and macrophages. So as you can see, this is the main structure, and the more outstanding achievement of the tumor cells during lymph node metastasis is that basically they are thriving in an environment that is populated by cells which in theory should detect and kill them. So as you can see, we know that, this is let's say a schematic representation, a very short and simplified schematic representation of the metastatic process for a lymph node. As you can see, with a normal lymphatic flow, the lymphatic vessel has a finite diameter, but then in the presence of the tumor, these vessels become dilated. This might have something to do with the increased needs of the tumor for oxygenation and blood and lymphatic flow. So when these vessels become dilated, the tumor cells sometimes find the opportunity to pass through and establish metastatic niches in the lymph node. This is a schematic representation, and here you can see what this looks like. First of all, macroscopically. and also microscopically if we label the cells with fluorescent dyes. So CD31 is for endothelial cells, and this is why we can see the diameter of the lymph node, and the GFP cells are the cancer cells, and this is from this paper in Science in 2019. So as you can see, we do know some of the mechanisms involved in the movement of the tumor cell from the tumor to the lymph nodes and in the establishment of metastasis, but of course, not all mechanisms are clear, and this is what we are attempting to study in this consortium. So the consortium has suggested the organ-on-chip technology as a powerful tool to study the interplay and crosstalk between the tumor and the lymph node. This is, of course, not a novel idea invented by us. The organ-on-chip market was already valued at 103 million in 2020, but it has an alarming growth rate actually of 30% from 2021 to 2030 annually. So it is projected to reach about $1.6 billion by 2030, and it aspires to provide many different physiologically relevant models, both for basic and translational research, and eventually replace in vivo experimentation. I personally think that we are still quite far away from this point, but if I do live to see the day that this happens, I'm going to be very happy. So as you can see here, you can see some of the setups that are currently available and some of the more ambitious setups that we hope to establish in the future as an organ community. I mean, this is a small chip with a number of wells in which you can place different cell types and examine their interaction, whether it be in real time with microscopy or with other endpoint analysis, as we are doing so far in our consortium, and as I'm going to show you in the next slides. Indeed, in the schematic representation of the organ-on-chip system here, you can see that there are people creating tumor organoids from stem cells, and this is something else that we are also trying to work on in the lab. I will not be talking about that today, of course. I will focus just on the lung slice culture. Just to show you some current organ-on-chip applications from works that have already been published, you can see that sophisticated setups are already in place and functional. Here is an example quite impressive for me of an intestine on chip. So you can see that people have actually even managed to reproduce the villus structures with the bottom of a vascular endothelium inside a chip. And here, where you can see strain, they even apply some sort of peristaltic movement to actually mimic the physiological movement of the intestine inside the human body. So they have media flow from the bottom of the chip, and from the top of the chip, they apply different types of bacterial stimuli. So this is actually almost exactly what happens inside the human body. This is a very good representation of a human intestine, and one of the most widely used applications of the organ-on-chip technology so far. Here you can see another chip which attempts to mimic the tumor on the chip, and this is just the tumor so far. So we have some endothelium, some epithelial cells, and the cancer cells on top of those. A similar setup to what we can see here. And this last image that I'm putting here is from certain groups actually attempting to recreate a lymph node on a chip, including some sort of extracellular matrix. In this case, they have gelatin and stromal cells at the same time as B cells, which work together to form a sort of immune organoid. And I'm including this because, as I'm going to tell you in our next slides, this is one of our more ambitious ideas that I have for this consortium. Well, you will see what I mean a little bit later on. So just to give you some information on the Tumour-LNoC consortium. This is the website for the consortium, and these are the links for some of our social media, just in case you guys want to follow us. And this is a very generic design, let's say, of the type of chip that we are imagining for this project. So as you can see, what we are proposing is to have one chip roughly the size of a microscope slide, so that in a more distal future, we can also attempt to follow movement of metastasizing cells in real time under the microscope. And we have designed, let's say, or well, our colleagues have designed 2 wells, one on each side of the slide. One of the wells is covered with a membrane that has pores that are 1 micrometer thick, and the other side is covered with a different type, well, with a different membrane from the same material, but this time the pores are 8 micrometers thick. Just to remind everybody, cells should be able to migrate through the 8 micrometer thick pores, but not through the 1 micrometer thick pores. And the setup that we have thought to apply in this case is to slice lung tumors fresh or through mice, and we will see why that might happen a little bit later. So slice lung tumors and place them on the membrane with the 8 micrometer pores, so that potential metastasizing cells can pass through those pores and end up on the bottom of the well. At the same time, on the other side, we are going to have lymph node cells, or maybe, if we are lucky enough, and this is what I want to attempt next with the Compresstome, whole slices of lymph node. Well, I don't know whether technically I will be able to do this, but I will definitely try in the next experiments that I'm going to do. So anyway, let's just say for the moment that we are going to plate lymph node cells in suspension here on top of this membrane. And as the pores are only 1 micrometer thick, the lymph node cells should not be able to pass through that membrane. However, they will be producing molecules which are, well, it has been shown in literature that draining lymph nodes in the vicinity of the tumor produce some sort of molecular cues that guide tumor cells towards the lymph node. So this is what we hope will happen also in this case. At the same time, lymph node cells will be, let's say, forbidden by the membrane to cross the pores and find themselves on the bottom of their well. However, this will not be the case for the tumor cells. Potential metastasizing cells will cross the membrane, and if they receive these molecular cues by the lymph node cells, they will start migrating through a tunnel that will be connecting the 2 different wells, and you can see this here. Of course, this channel will be at the height of the wells and not at the height of the membranes. Okay, so this is going to be just below the membranes. Just to make this a little bit easier for you to imagine, this is a side view, let's say, of the setup that we are hoping to achieve. So on one side, we have the 8 micrometer thick pores with the tumor slice on top. And on the other side, we will plate the lymph node cells on top of the 1 micrometer thick pores membrane. And we hope to see metastasizing cells traveling from here through here. And our ambition is to manage to keep this system viable in culture for at least 1 week. Like I said, this is a generic chip design. Once the design of the chip is optimized and completely finalized, and our colleagues from other groups are currently working on this, as you can see here, there's going to be some inlets and outlets, and we hope to connect to a microfluidic device that will be pumping fresh media all through the culture. We also hope to achieve different media compositions for the tumor and for the lymph node cells at a later time point. And like I said, our colleagues from the other groups are currently working in optimizing this microfluidic circuit. There are many physical, let's say, considerations to think here. Some of them being, for example, that we need to completely seal this from the outside air to avoid the passing of air bubbles in our circuit. When these membranes are when the pores of the membranes have a different diameter, there is different flow in one chamber than the other, and we are trying to solve this because the flow should be similar, just to be sure that it's not the flow of the media that is taking the cells from one chamber to the other. And well, like I said, our colleagues, the engineers and the physicists are currently working on this, And when it's optimized, we are going to use this with our tumor slices and our autologous lymph node cells that we receive from the same patient at the time of surgery. In the meantime, we are, of course, performing experiments trying to establish that we are capable of producing viable slices from lung tumors and keeping them in culture in the presence or absence of lymph node cells for at least a few days. Like I said, we are hoping for a full week of culture. These are some of the tests that we have performed initially. And this is where I'm going to start talking about how we use the Compresstome. As you can see, we are using the very well characterized system of transwell inserts. We have some transwells with the membranes that I indicated previously. So some of our membranes have pores of 8 micrometers and some other membranes have pores of 1 micrometer. And we are currently just using the 1 micrometer pore membranes as controls, just to make sure that no debris from the tumor tissue or no cells pass through those pores, and that we will indeed be able to use those as a control in our chips. How do we go about slicing up lung tumors? So I'm going to use a phrase now that I'm sure that most of us really hate when we see in the literature, but in this case, it is absolutely true. As you can see on this slide, we pretty much follow the provider's instructions. So when I received the Compresstome instrument, I also received the training that Precisionary instruments have designed for this. So I watched the video and I read the specifications very carefully. And as I'm going to tell you in some of the next slides, I just do everything exactly as Precisionary suggested. And so far, I'm very happy with what I'm getting. We are indeed managing to obtain uniform slices of a finite thickness and to maintain them, well, viable also, of course, and to maintain them viable in culture so far for at least 3 days. Just some technical details for our protocol. Like I said, we receive the tumors immediately after surgery, and this is one limitation of our work, because of course we can only receive what the pathologist doesn't deem is necessary for them for diagnosis. So we get the spare material. There is not often that much spare material, but we, as I'm just about to tell you, we have found a way of dealing with that as well. So we receive 1 chunk of tumor tissue, some of the patient's blood, which we use for the culture, and one of the lymph nodes, or a chunk of lymph node that the pathologist feels that they can spare, and they send this to us. So we transport the tissue and the lymph node in calcium and magnesium-free HBSS supplemented with some antibiotics. And if the tumor chunk is large enough and 3D enough, then we immediately process for Compresstome slicing. By 3D enough, I mean that sometimes from some of the tumors, we do obtain quite large tumor chunks, but they look more or less like membranes. And in that case, there's really not much that we can do. However, there is one thing that we can do to facilitate, let's say, our work. So if the tumor chunk is too small or too flat, we first implant it in mice, and we wait until it grows an adequate size. So basically, we establish a patient-derived xenograft inside an immunocompromised mouse. This is a well-characterized process and widely used in the literature. And when the tumors grow enough, well, first of all, we have the possibility to passage them in mice, so we can keep tumors in the lab from one patient for a longer time frame. And when it grows an adequate size, we then process for Compresstome slicing. Meanwhile, we have also stored cells from the lymph node in suspension. So from one human lymph node, we can get as many as 200 million cells. So we can freeze them in many different aliquots. And while keeping the tumor alive with the PDX technology, we can perform many experiments with tumor from the same patient and autologous lymph node cells from the same patient. So what we usually do to prepare for slicing is that we cut the tissue in chunks of roughly 1 by 1 centimeter. And we prepare low melting agarose in a percentage that Precisionary themselves suggest for, let's say, spongy and softer tissues, right? So 2.5% agarose works pretty well for us. And as you are going to observe in the video, this has an added advantage that I'm going to tell you all about. So we glue the tissue on the specimen tube and embedding the agarose, and we slice this in ice cold PBS with some antibiotics. As per Precisionary's specification, we use a relatively low oscillation and speed because the tissue is not fixed, it's fresh, and it's of a relatively smooth mother tissue, let's say. So let me just show you here how the Compresstome works during this process. Okay. So please note that while the slice is detaching itself from the main block, you can see that the tissue slice is practically almost already detached from the agarose. For us, this is an advantage, and indeed Precisionary in their instructions warn us that this will happen. This is another reason why we choose a relatively high percentage of agarose for embedding. So this is already pretty much detached from the agarose, and this facilitates very much my work when I have to put this in culture, because I do not put the tissue slice in culture in the presence of agarose, but I detach it first. And here is how I harvest these slices. So I just use a microscope slide, and with some careful handling and with the help of the forceps provided by Precisionary, which are, by the way, I think very, very good and very adequate for this type of work. They don't damage the slice or the agarose at all. They are some of my favorite forceps that I've ever used. So there you go, the harvested agarose tissue slice looks like this. And when I have this, let's say, fished out of the buffer tank, I just dip this microscope slide in a 50 ml Falcon, once again with PBS supplemented with antibiotics, and I store all my slices in the same Falcon until such time as the slicing procedure is finished. So, okay. So just some considerations regarding protocol. I have found that it is better to embed the tissue deep enough in the agarose to ensure maximum compression upon slicing. What do I mean with this? So you will see that even in the instructions provided by Precisionary, because this is from the Precisionary website, this here. And let me zoom in a little bit so that you can appreciate that this tissue is placed. So in the beginning, I was trying to have as little agarose as possible on top of my tissue, so that of course I would not waste so much time with the blade, because keep in mind that this needs to be a process that is as fast as possible. So just, I mean, I was trying to get the blade to the tissue as quickly as possible. However, keep in mind that when agarose is still at the brim of the specimen tube, compression is not at its maximum. So the first few slices that I was getting were not very, very good. So if you will notice here, the entire tissue is below the point where the diameter of the tube becomes the widest. And this ensures that when the blade encounters the tissue, we will have the maximum compression to ensure uniform cutting from slice number 1. This is very important for me because, like I said, the tumor chunks that I receive from the pathologist are not always 3D enough. And sometimes I have tumors as short as ½ centimeter. So meaning that, I mean, of course, I will lose some on the basis of the specimen tube where it's attached to the glue. So I really have limited thickness, let's say, from which to acquire a number of slices. So yes, this is, I find this to be quite important. I have also observed that, well, in my hands at least, I set the water bath at 42 degrees to cool down the low melting agarose a little bit before, of course, putting it inside the specimen tube, because if it's too warm, if it's too hot, it's going to damage the tissue. So I keep it there for about 5 minutes. 5 or 10 minutes, and then it's safe to use. But I have noticed that it will solidify if it's kept at 42 degrees for longer than about 10 minutes. And this is a bit of a nuisance for me, so I have to prepare it again. If any of you have any suggestions on this, please let me know. I also wanted to mention that between the magnetic blade holder and the one where you need to glue the blade, I have found that the magnetic blade holder is more convenient for me, and it works quite well in my hands. I regularly change the buffer in the tray to make sure that it is indeed ice cold. So I change the buffer in the tray with freshly chilled buffer approximately every 15 minutes. I, of course, use a new blade every time a new tissue arrives. And a very important trick that I've learned through bitter personal experience after, well, vibrotome slicing. I always keep and store the glue tube on an upright position, both during the use and during storage. I make sure that the tube is always upright, because otherwise sometimes the glue solidifies at the brim of the tube and you can no longer use it. And in my case, this would be this would really be a pity, because Precisionary glue is one of the best glues I've tried for this purpose. So for the tissue culture, I just wanted to tell you a little bit how I do it. And I will also use some of my props to actually show you what I mean. So in order to keep the slice as undamaged as possible, I try to avoid picking it up with forceps. And for the moment, I have to place it inside a transwell, which has some depth. Okay, so for this is the type of transwell that we use. And you will see that this will fit a 12-well plate. Okay, so we can put a relatively large slice of tumor in here. It can have a diameter of up to 1 centimeter, maybe a little bit more, but it has a depth. So this is what I use to place the tissue on the transwell membrane. As you will all immediately observe, there is a conflict here because this will not fit the transwell. So what I end up having to do is under the hood, I will use some really good pairs of scissors to cut this to the dimension that is more convenient for me. And as you can see, it will now fit the transwell. I have also observed that it is very uncomfortable to try and put and place a slice immediately on the transwell membrane. So what I have found works best for me is to fill the transwell with some media and just dip the cell lifter inside the transwell. The slice, upon encountering the wet media, will immediately float on the media. It's going to be completely flat. And so then I just have to aspirate the media from inside the well, and I end up with a transwell insert with a very flat slice on top, which I have handled minimally in order to try and get it there. So yeah, this is how I go about placing the slice on top of the membrane. And then I place this insert in a well with media in the presence or absence of lymph node cells. So one of the first biological questions we want to answer is whether we will observe more metastatic activity in the presence or absence of lymph node cells. So what we are hypothesizing based on the literature is that this will happen more when lymph node cells are present on the bottom of the well. And I don't really have statistics from many experiments yet to share any actual data with you, but I can tell you that the first indications from 2 or 3 independent experiments that we have run is that this is indeed happening. So when we place the tumor on 8 micrometer thick pore membrane, we do have significant migration to the bottom of the well, even in the absence of lymph node cells. However, when lymph node cells are present, we observe a higher number of tumor cells passing through the well. We cannot really explain this molecularly yet, but that's exactly what we aspire to do in the course of this project. We change media every other day. We use the media described in this paper here, Nature Communications of 2014. And according to this paper, tumors were more viable if the media was supplemented with 2% from autologous sera from the patient. And this is why I mentioned earlier that we have asked our collaborators, the surgeons, to also provide us some of the patient's blood. We have performed some tests and we have seen that indeed this is the case. And after some days, we perform endpoint analysis. So in our case, we do histology to check the integrity of the slice, and we do flow cytometry to try and check, well, like I said, percentage of EpCAM positive tumor cells on the bottom of the well and CD45 positive immune cells coming from the lymph node. This is what we have done so far. We hope to be able to take this one step further later on and perform a more detailed characterization of how the lymph node cells behave when the tumor cells pass through the membrane. We do, of course, also place one control condition on which we only include lymph node cells at the bottom of the well. And this I can tell you with absolute certainty. After about 48 hours, most of these cells are in very bad conditions, the lymph node cells, I mean. Most of the immune cells are in very bad condition. They are almost all apoptotic. This is to be expected. Primary immune cells do not survive for a very long time in culture unless provided some kind of stimuli. But when a tumor slice is placed on top of the transwell insert, this does not happen. Lymph node cells remain much more viable. And we also have some indications that populations that are generally very, very sensitive to cultures, such as macrophages and dendritic cells, persist for a number of days in the presence of the tumor slices. So now for some of our future plans, we would like to further optimize some of the technical aspects. And I'm saying this because after every experiment, we check with the pathologist with whom we collaborate and who provides the actual tumor samples. And we check whether, so first of all, whether after Compresstome processing, the slices were actually viable, and whether after the number of days that we leave every culture in every different experiment, so whether those slices still continue to be viable at the end of our experiments. And we have seen in some cases that some tumors do not respond well to the process that we use for the Compresstome slicing. We don't know what might be the problem about this yet, but we are trying to figure out alternatives, and whether it's the temperature from the agarose, whether some tumors are just more sensitive to any type of treatment as it is, and but we are working to further optimize the protocol to try and obtain viable slices, well, pre-culture slices from all the tumors that we will be receiving. We would like to increase our statistics so as to have actual data on all the preliminary concepts that I mentioned in the previous slide. Like I said, one of my more ambitious ideas for this project is to attempt to slice whole lymph nodes and actually culture on our chips tumor slices and lymph node slices, so tumor slices on one side and whole lymph node slices on the other side, so as to have the entire structure that we saw on some of my first slides, so like the endothelial cells and the stromal cells and everything. So ideally, we would also like to successfully set up the organ-on-chip protocol with a flow, so providing fresh media to both the tumor and the lymph node all through the culture, rather as opposed to changing it every other day, which is what I'm doing currently. And ultimately, what we have promised the European Union that we are going to do with this, to characterize the metastasizing cells at the molecular level and compare them with non-metastasizing cells. So I have to acknowledge everybody else that really works very hard on this project. I want to start, of course, with the lab where I'm currently working. Of course, our principal investigator, Apostolos, who decided to give the Compresstome a chance when I mentioned that such an instrument exists. Anna Halari, that is, who is doing all the work with the PDX establishment, and my PhD student Christos, who is always there to lend me a hand when we receive a patient sample, and I'm busy with the Compresstome, but there are also like 3 or 4 other different things to do at the same time. The Tumour-Lymph Node on a Chip Consortium, we really have some amazing collaborators there. We are very lucky to be a network of many labs across Europe with many different expertises. It's a very interdisciplinary project, and I feel really lucky to be a part of this. The coordinator, Ioanna Zeriotti, and Maria, who is trying to set up the microfluidic circuit and make it work flawlessly with no leaks, no air bubbles, and no nothing. And this is proving to be a very challenging enterprise. The BRFAA animal facility and the histology facility, as well as our, of course, the collaborators in the Sotiria Hospital, a hospital that is, I think, a world reference point for thoracic interventions and you know, like lung health. So surgeon Konstantinos Potaris, pathologist Yannis Vambakaris, and specializing Dr. Georgina Zahu for providing all the samples and for bearing with us every time we need to consult them on whether whether another one of our experiments worked or not. And of course, thanks very much to all of you for your attention. I am ready to take your questions or suggestions or any other comments that you might have.
Wonderful. Oh my gosh, Katerina, thank you so much. And for showing us your tumor slicing strategy, your tumor on a chip, and also the videos showing how you collected the tumor slices. I definitely wanted to open up the floor and see if anybody had any questions. Feel free to enter your questions in the chat or just unmute yourself and ask. To kick us off, I had a quick question for you, Katerina. Is you mentioned that sometimes it's very advantageous for your experiments to keep the agarose like that rim around your tissue, your tumor slice, yes, even though you want to minimize that agarose as much as possible and maximize compression. In cases where did you have any cases where you do want to remove that agarose? And if you did, do you have any strategies for how to get your slice out of there without damaging it?
Yeah, so like I said, well, so the guide from Precisionary mentioned that the thicker the agarose, the easier it's going to be to detach it from the tissue. And as you saw in the video, so if that's what you mean. So as you saw in the video, was that your question?
It was, yes.
Okay, so as you saw in the video, with a 2.5% agarose mix, at the moment of slicing, what happens is that the agarose itself just sort of automatically half detaches from the tissue. What I do, if it doesn't detach fully, what I try to do is that, like I said, I harvest all of my slices in a 50 ml Falcon, and then I just deposit them in a 10 centimeter, in a normal 10 centimeter Petri dish, like, you know, like just like the ones we use for tissue culture. So then what I do is that I will reharvest one of those slices with a microscope slide, and I will gently deposit it on the plate on the plate top, and then just use the Precisionary forceps and some scalpels. To remove the rest of the agarose. But when the agarose is at 2.5%, so when you use a 2.5% agarose mix, this is really easy. And I will go about just touching the agarose with these forceps, which, like I said, are fantastic for this. And if I ever have to touch the actual slice, these are the forceps to use. So I try to, if I absolutely need to touch the slice, I try to touch it just with a forceps and not with a scalpel, and just touch the agarose itself with a scalpel. But generally speaking, if you use a 2.5% agarose mix, this is a relatively easy process.
Got it. Got it. Wonderful. Does anybody else have any questions? And if not, I'll wrap things up and say thank you to Katerina, our speaker, and to all of our attendees. Thank you so much.
Thank you. Thank you, Abby. Thank you for having me.